or so. Thanks. All right, I think we're going to get going. So hello and welcome everyone to our weekly journal club. My name is Jonathan Nasi and I'm the Director of Translational Science at Inscopix. We started this series at the beginning of April to help keep the scientific community connected during these difficult times. We'll be periodically discussing publications of importance to the field of circuit neuroscience, often featuring Inscopix Envista or Invoke Imaging, and we're excited to have you join the discussion from your virtual offices at home. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation is over. We'll do our best to get to most or all of the questions with a hard stop right at 12 p.m. Pacific time. Today, our field scientific consultant, Giorgio Rizzi, will be presenting a Frontiers in Cellular Neuroscience publication from Mark Baer's group at MIT. Giorgio received his PhD at the University of Basel in Kelly Tan's lab, where he went on to do a postdoctoral fellowship as well. Over his time there, he studied the neural circuits underlying nociception, dyskinesias, and other motor behaviors, gaining experience in a range of techniques, including electrophysiology, optogenetics, and calcium imaging. And with that, I'll hand it over to Giorgio. Thank you.
Thank you, Giorgio. So good morning or good evening, everyone, depending on where in the world you're joining us today from. So today I'm going to present a paper from Mark Baer's lab at the Picauer Institute for Learning and Memory at MIT, entitled Opposing Somatic and Dendritic Expression of Stimulus Selective Response Plasticity in Mouse Primary Visual Cortex. So a little bit to unpack on that title. So let me start with some background and with some terminology that is going to be useful for the rest of the paper. So the primary visual cortex, or V1, expresses experience-dependent plasticity that serves as a form of recognition memory that alters behavior. So what that means is that the visual experiences that you can have are able to induce lasting changes in the brain and how neurons respond to certain visual stimuli. And this actually ends up biasing behavior or changing your behavior. So for example, as a previous study in 2015, Cook et al., they showed that if you take a mouse and you put it in an open box with two screens on the sides, and you present a visual stimulus, in this case, it's a graded stimulus where you invert the faces of the screen. Once the stimulus is novel, the mouse will approach it and will explore this area. It will spend more time on this area, and as the stimulus is represented over days, the mouse becomes habituated to the stimulus, it becomes familiar, and now this preference is lost. And you can, without going through the entire figure, you can see, for example, in panel D of this study, if you look at the preference for the stimulus presentation zone, on the very first day, there's a higher preference, and as you expose or habituate the mouse, the amount of time spent in this area of the box goes down. And if you change the direction of this grading, so you present a novel stimulus, then this behavior comes back again. This novelty exploring behavior comes back, if you will. So for example, if you were to compare the stimulus zone preference between the novel stimulus presented and the familiar stimulus, you will see that there's a much higher preference for novel stimuli. So this, in a nutshell, so phase reversing stimuli induce orientation selective habituation, or OSH, and this triggers visually evoked potentials. So these are field potentials that are generated in the primary visual cortex that respond to this particular orientation stimulus. And as you present this stimulus on repeated days, as the behavior is habituated, these VEPs, or these field potentials, increase in amplitude. So if we look at Frank et al. In 2006, for example, here you can see how if you measure these VEPs or the amplitude of these VEPs from day 0 to day 4, you can see that they grow with time. So you have a behavioral habituation and you have an electrophysiological substrate of that habituation. It is also of note that the VEPs that you can record from the contralateral eye are about 3 times bigger than the ones you get from the ipsilateral eye. So as OHS develops and develops VEPs to a familiar stimulus, as OHS develops, VEPs to a familiar stimulus grow, and this phenomenon is called stimulus selective response potentiation, or SRP. And that's going to be the center of this study. As an added note on this plasticity phenomenon, it is dependent on NMDA receptors in V1, and it's also disrupted by ZIP and local manipulations of PV interneurons. So even though you could imagine that the behavioral response or the behavioral adaptation involves several circuits in different areas of the brain, it is interesting to note that a local plasticity happening in V1 is necessary for the expression of this phenomenon. So, as we get into this study, we have the background, we have the phenomenon that is very well known and characterized. What this study tackles is what's happening at the single cell level. So it is known that when you record these field potentials, when you stick an electrode in the cortex and you measure the responses to a specific orientation stimulus, these responses are growing, but it is unknown what is happening at a single cell level. And to do this, the approach of the lab was to get a triple transgenic mouse line that would express GCAM6F in EMX1 positive cells in the cortex in a conditional manner with the TTA system. So essentially, they cross transgenic lines and they make sure that the expression, the TTA system, is only activated after weaning, so after P21, to make sure that the normal development of the visual cortex is happening. And then after this, they go on to do all their experiments. And as a first step, what they do is they do a control experiment to make sure that expression of GCAM in these EMX1 positive cells in the primary visual cortex is not altering this plastic phenomenon. So in panel A of the first figure, you can see an example representation of the GCAM expressing cells in V1. As a control, you have a counter stain for parvalbumin, that is a typical marker for interneurons in the cortex. And you can see that the overlap between the two is very minimal. So this confirms selective expression of GCAM on principal cells in V1. Now, they take these mice, they put them through a paradigm where they go through 2 days of habituation, looking at a gray screen. They're just getting used to being restrained. And then through days 1 to 5, they are presented phase inverting stimulus at a specific orientation, and they record the VEPs throughout this whole process. And what you would expect, because what we just saw, is that as days go by, the VEPs elicited by the stimulus should be growing in V1, and you should have a stronger stimulus coming from, or a stronger VEP in response to the visual stimulus coming from the contralateral eye. And this is in fact what they see. If we look at panel C, when they look at the magnitude of VEPs, You can see, first of all, that the contralateral VP is stronger than the ipsilateral, both in control mice and in transgenic mice. And more notably, there is no difference or no statistical significant difference between the control mice and the transgenic, either in the contralateral side or the ipsilateral side. Now, they also looked at the sensitivity of the V1 in these mice, depending on the contrast of the stimulus they present. So they go from low contrast to 100% contrast. And as you can look at the normalized VPs as percentage of baseline, the distribution between the transgenic mice and the control mice is pretty much identical. The same thing goes for the visual acuity. So if you test the spatial, or you increase the spatial frequency in cycles per degree, so the distance between the bars in the stimulus is getting finer and finer, the normalized VP percentages of baseline is pretty much distributed equally between control and transgenic mice. When you look at the VPs, from day 1 to day 4, and then when you switch the stimulus on the 5th day so that you go to a novel stimulus, again, the progression of the magnitude of the VPs is pretty much what you would expect, which means growing as the stimulus becomes familiar and going back down as you present the novel stimulus. They observe this both in control mice and in transgenic mice. And if you were to take, obviously you cannot directly compare them because they're different groups of animals, but if you were to calculate the ratio between the novel stimulus elicited VP and the familiar stimulus and the novel stimulus, you will see that the ratio between them is pretty much identical between control and transgenic mice. So this is a really nice first step to check that everything is happening in the cortex as it should, and that particularly the expression of a calcium buffer protein in these principal neurons in V1 is not changing the properties of neurons and is not altering this plasticity. So next, the authors wanted to check at the distribution of cells that respond to these visual stimulus. So as we know now that the VPs are elicited and they increase with time, one of the hypotheses is that you could potentially have more cells being recruited and that are now responding or start to respond to this particular visual stimulus. So to test this, the authors now took their mice, they put them under a 2-photon microscope, they presented the screen in front of them, and they pretty much go through the same protocol, which is pick a specific visual stimulus and then alternate that with gray screens to run in a semi-randomized way, and then look at the VPs, or in this case, look at the activity of principal cells and see whether they are being activated or not throughout the whole presentation of the stimulus. Now, it's important to keep in mind that this particular experiment is looking at the activity of cells throughout the entire presentation of the graded stimulus. And here the authors, they took a couple of different criteria to figure out how to establish what would be a relevant calcium transient or an event. And essentially what they did is they took 15 ROIs from the background of the field of view, and they average the signal from these 15 background ROIs, and they take 3 times the standard deviation of this baseline, if you will, and any signal above those or outside this 3 times standard deviation is considered an event or a relevant event. That's one of the criteria. On the other hand, in order to decide which cells to include as being active or participating in the neural representation of this visual stimulus, they went from cells with at least 1 calcium event during the presentation of the stimulus to at least 20 events during the presentation of the stimulus. And this is what you see in panel B. So this is the data regarding at least one event, at least 5 events, at least 10 events, and 20 events. And essentially, regardless of how many events they request from the cells to consider that they are being activated from the stimulus, they saw no difference in the fraction of active cells from the first day to the 4th day of presentation of the grid. And the same thing goes for the presentation of the gray screen. And these happen both from day 1 to day 4, and also on the 5th day when they test, if you will, the mice from the presentation to the familiar stimulus to a presentation of a novel stimulus. And this pretty much happens regardless of whether you pick 1, 5, 10, or 20 events. Of course, you can imagine that as you increase your threshold, so as you request a higher number of events for your cell to be considered as being active or as being or activated due to the visual stimulus, you will see a lower fraction of active cells. But when you compare day 1 to day 4 and familiar to novel, there was no statistically significant differences. The only one thing to note is that when you look at the presentation of the gray screen, on the 5th day, when you go from a familiar stimulus, so a gray screen right after you just presented a familiar stimulus, to the presentation of a gray screen after you just presented the novel stimulus, there is a statistical significance once you start looking at particularly active cells, so cells that had at least 10 or 20 events, which could be an indication of increased spontaneous activity on day 5 after you have habituated the mouse to that particular stimulus. Now, in order to check whether maybe 3 times the standard deviation was a criteria that is too strict and you perhaps are filtering out other events that are meaningful for this process, they also decided to lower that threshold to 2 times the standard deviation. But even lowering this threshold, you can see that the fraction of cells between a familiar and a novel visual stimulus was pretty much identical. So even if you were to be more generous and then just capture smaller events, there was no statistically significant difference between the moment you present a familiar stimulus on day 5 versus when you switch the orientation of that grading and now you present a novel stimulus. And if you look at the mean event number with 3 times the standard deviation, there is no difference between familiar and novel. And if you go to 2 times the standard deviation, of course, you have more events, but there's no difference between familiar and novel. So interestingly enough, this excludes that first hypothesis that the increase in VEPs throughout the days, like from day 1 to day 4 or 5, is due to recruitment of more cells in V1 that start encoding these visual stimulus. This pretty much shows that this is not the case, so something else must be happening. Now, one thing that I mentioned at the beginning is that the calcium imaging taking place in that previous experiment was done throughout the whole presentation of the stimulus, and especially throughout the whole process of inverting phases. Now, the VEP is a potential that is induced 50 to 150 milliseconds after you invert the phase of this grading. So one hypothesis here is that perhaps you need to look with a much finer temporal resolution to be able to detect any changes or any adaptation happening with these cells. And for this is where Inscopix technology comes in. They use, so before I forget, the limitation of doing that with the 2-photon system is that their system was limited to a 4 hertz sampling frequency. So this, of course, is not fast enough to detect those 50 to 150 millisecond intervals. So that's why they switched to an Envista system from Inscopix. And in this case, their strategies changes a little bit. So instead of using the triple transgenic mouse line, they go even more specific and they use a SCNN1A Cree mouse line, which is specific for layer 4 neurons in V1. And they use a Cree-dependent GCAM6F virus. So they inject the virus in V1. They use a prism lens inserted in V1 to be able to image these neurons. Over here in C, you can see the track left by the lens and a really nice selective expression of GCAM in layer 4. So in terms of behavioral paradigm, they also change things a little bit here. The first part is pretty much identical. So you have 2 days of habituation where the mouse is just getting used to being head restrained and it's just seeing a gray screen. And then from days 1 to 4, it's presented the same visual stimulus and it's supposed to habituate to it and the stimulus is supposed to become familiar. On the 5th day or the test day, this stimulus is presented, and right after that, a novel stimulus is presented by switching or rotating the orientation of this grading stimulus by 90 degrees. And then this novel stimulus is presented for 4 more days so that the mouse becomes habituated to that one. And then on the 9th day, the same test paradigm takes place. They present this x degrees plus 90 novel stimulus that has now become familiar. And as a novel or as a second novel stimulus, they present the same pattern rotated by 30 degrees in relation to the original stimulus. So in here you can see the response of calcium imaging of cells in relation to the inversion of phase. So this would be phase 1, and this would be the inversion phase, and then you go back to the phase 1, so on and so forth. And then on the y-axis you have the cell numbers. So in this case we're talking about 120 plus cells. And essentially as they look at day 1 to day 5, and they look at the calcium transients that they can observe during these phase inversions, you can see that when the stimulus is novel, so on the very first day, there's a fairly strong response in terms of calcium transients. And as you habituate this response, it goes down. Now on day 5, you can see that it's significantly lower than on the first day. So novel stimulus has a smaller response in terms of somatic calcium transients. And when you rotate the stimulus, then you have stronger calcium responses. And as you would expect, during the second habituation phase, these responses go down. And on day 9, once you rotate once more, so you represent a second novel stimulus, then these responses are stronger. So that's quite interesting if you think about how it compares with the VEPs. So this is the summary data. So when you look at the mean delta F over F from day 1 to day 4, it significantly decreases. And on day 5, when you test these mice, it significantly increases. So essentially, novel stimuli induce stronger calcium responses in layer 4 neurons in V1, in the primary visual cortex. And this is true whether you look at, or whether you take a 50 to 1200 millisecond window after the phase inversion, or if you take a much narrower window and you just look at that 50 to 150 millisecond window, which is what the usual VEP time window would be. So regardless of whether you take a larger or smaller sample window, you pretty much see exactly the same thing, which is strong responses to novel stimuli, weaker responses to habituated or familiar stimuli. So that's quite interesting because you can see that it's pretty much the opposite of what you see with VEPs, right? With VEPs, you would have a relatively small response with a novel stimulus, and as it becomes familiar, then these potentials are growing with time. So that's a question that the authors are gonna answer in the rest of the study. So before we get to the explanation of that, another interesting thing that the authors looked at was how these calcium responses are adapting, if you will, inside or within each block of visual stimulus. So essentially, like I mentioned before, the paradigm consists on the graded stimulus alternated with gray screen. So what the authors did was to look at the very first phase inversion compared to the very last phase inversion within each stimulus block presentation. And you can see that when you compare the first day and the fourth day, so when you look at novel versus familiar stimulus, you can see that the biggest difference throughout the blocks is at the later phase of the block. So you can see that, first of all, there is some adaptation, and the largest difference from day 1 to day 4 is happening during the last phase inversion of each block, which means that even at this short time scale that don't require, is not hours, it's just minutes, there is already an adaptation process happening in these V1 neurons. If you look at on day 5 at the familiar and the novel stimulus, you pretty much see the same thing. So at the later phase of each block, so towards the last phase inversion of each block, is where you see the biggest difference. And then over here you have the summary data. So essentially, if you look at the mean delta F over F, when you compare in day 1, the first versus the last phase inversion, there isn't a strong change in that, which means for novel stimuli, there's not much of a change within each block, but for familiar stimuli, there is a lower response to the last phase inversion. When you look at day 5, when you compare the familiar stimulus and the novel stimulus, you pretty much see the same thing. So when you look at the familiar stimulus during the last phase inversion, the responses are smaller than the first phase, and with the novel stimulus, they pretty much remain high. There's no adaptation happening there. So if you take then what I said previously and do the direct comparison with VEPs, then you pretty much have a mirrored image. So the authors, to confirm or to understand what's the substrate of these seemingly opposing findings, they go back and they record VEPs on these mice. They put them through a very similar paradigm. I think they add a couple of habituation sessions, and they also increase the number of blocks or the number of phase inversions that they present to the mice for one simple reason. VEPs are potentials that are pretty much mounted on EEG activity, which means that things are going to be very noisy, and usually you average across trials, if you will, for each mouse, and you're able to filter out that EEG or any background noise like that. In this case, because they want to look at each block individually, and especially at the first phase inversion versus the last phase inversion, what they do is they average across animals, and to be able to reduce a little bit more the noise, they just increase the number of repetitions that the mice are exposed to. The data in panel A and B is pretty much the same analysis that they did before. They're comparing the first phase inversion versus the last phase inversion throughout the blocks, comparing day 1 versus day 4, and the familiar versus the novel stimulus on day 7 this time. If we go straight to the summary data, then for for novel stimuli, there is a drop in the response from first to last phase inversion, even though it's not statistically significant here. Whereas for familiar stimulus, it remains the same. On day 7, when you compare the familiar stimulus and the novel stimulus, it's pretty clear that with a familiar stimulus, there is no adaptation, whereas with a novel stimulus, there is adaptation. So the experimental conditions are right, what the observations are correct. If you look at the previous data, so maybe if I quickly go back, you see with the panel D and E are pretty much the mirror image of panels C and D here. So when you look at somatic calcium transients, there is no adaptation for novel stimuli, but adaptation for familiar stimuli. When you look at VEPs, there is adaptation for novel stimuli, but no adaptation for familiar stimuli. And this is pretty much the big question to reconcile up until here. Like this, I think it was quite unexpected for the authors. And their hypothesis was that perhaps the activity that is being measured from VEPs. So if you think about what a VEP is, which is pretty much an extracellular current that you're picking up with an electrode that you stick in the brain, that is coming from radially aligned dendrites in layer 4 of primary visual cortex. You can do I forget the name, it's a current source analysis, and this pretty much shows that these VEPs are derived from depolarizing currents in layer 4. So cells should be being depolarized, and this is what induces these currents. So on the calcium imaging side of things, you should be seeing increased responses, not decreased responses. Now, their hypothesis is it's probably happening in the dendrites, but it's not transferring to the soma. So what they do is they go back to those background ROIs that they had selected before, and they analyze that background signal. And essentially what they find is that when you look at the mean delta F over F of this background light, it starts relatively low, and as you habituate the animal, or as the stimulus becomes familiar, it increases. So this calcium activity coming from neuropil is following what VEPs are actually doing. And the same thing goes for familiar versus novel stimulus on day 5. So with the familiar stimulus, you have a stronger fluorescence, and as you present a novel stimulus, it becomes weaker. So, yeah, essentially here, then the authors confirm that there is this opposing mechanism or opposing phenomenon happening in layer 4 of the primary visual cortex, where as you present a visual stimulus and it becomes familiar, and there is a behavioral habituation that comes with it, there is actually an increase in dendritic activity. So there's a stronger input coming into those dendrites of layer 4 principal cells, but there must be a strong shunting inhibition that is suppressing the soma activity, so that it's not resulting in in action potential being driven downstream. Now, as the final step of this study, the authors wanted to check whether the induction of SRP changes the orientation tuning of these layer 4 cells. So essentially what they do is they take these mice and they probe the response of each cell with different orientations of the grading, and these cells usually have a preferred orientation, which is the phenomenon called orientation tuning, and they test this before and after induction of SRP to see whether it's changing or not. Now, the authors do so in order to not influence the subsequent induction of SRP by presenting the stimuli. What they do is they perform this test in the presence of CPP, which is an NMDA antagonist. And like I said in the introduction, an NMDA antagonist will block the expression of this plasticity, allowing them to test the orientation of the orientation tuning of these cells without inducing or without occluding the subsequent SRP induction for their test. So in panel B, you can look at the orientation selectivity index that they define for the study, and you can see that before SRP and after inducing SRP, the orientation selectivity index is pretty much identical. So the plasticity that is taking place in these cells is highly selective or exquisitely selective to that one particular orientation of stimulus. It's not affecting the tuning, it's not recruiting more cells to respond to that particular orientation. And this is also shown in panel C, where they look at the fraction of cells as a percentage of the total number of cells for the particular orientation tuning that they're going to use, or orientation that they're going to use to induce SRP. You can see that it doesn't change before and after SRP, and it also doesn't change for the rotated novel stimulus before and after inducing SRP. And then in panels D and E, they have the distribution of the responses of cells between day 1 and day 4, organized in the same way, and day 0 and day 5. So from the first to the 4th day of presentation of the stimulus, and also from the day before to the day after the SRP. And this shows that there is no apparent effect on the tuning properties of these neurons. They are only changing their response specifically to that one particular visual stimulus that is being presented from this 1 to day 4. So this is the last figure of the paper. I think it's a very interesting study. I think it's very thoroughly conducted. It was definitely unexpected, and I think the authors did a really good job figuring out how come at the beginning they were seeing seemingly opposing phenomenon that, you know, when you're in the blind, you could start questioning whether what we're observing with calcium imaging has anything to do with what we're observing doing electrophysiological recordings. And I think it's a pretty good example of there is no one perfect technique and there is no one truth. And it's a very good idea to combine different approaches and use a multidisciplinary perspective on things to be actually be able to understand what's happening in the brain and how the brain is encoding all the information that is taken in from the outside. So with that, I think we can check if we have some questions.
Great, thanks, Giorgio. Appreciate that. Let's see. I think some questions are coming in. I'll just kind of reiterate what you were just saying, which is that, I do think one interesting aspect of this study was, there aren't too many studies as of yet that have sort of tried to relate macro scale kind of readouts, macro scale EPHIS like VEPs, and more mesoscale, microscale measures such as cellular calcium imaging. And I think there's definitely like a lot of interesting work to do there. And actually, one thing they mention, I don't think they do these measurements in this study. I'm not sure if you mentioned it in your overview, but they do also talk about some previous work where people have looked at just single unit spiking activity, which from these cells within this paradigm. And from what I remember, they do see very short latency increases in multi-unit spike activity, which, again, at first blush kind of makes you even more kind of scratch your head and kind of wonder, well, there is sort of the neuropill versus somatic calcium story and how the neuropill signal may relate more to the VEPs, but then you look at the short latency multi-unit spike activity, that seems to increase as well with familiarity versus the somatic calcium activity being reduced. And I think the authors explain that potentially that the calcium signal being slower is maybe, not necessarily catching that very short latency increase in spiking, but that what you're picking up more is a slower polysynaptic inhibition that might be involved in this kind of processing. So, but it just gets to the point that yeah, there's really interesting to kind of study phenomenon like this at these different levels, and even with the calcium signal, you don't too often see people look at the somatic versus the neuropill signal, and there are different things you can pull out from those different aspects. So I thought that was made it a pretty unique paper, and be interesting to. I think they mentioned, combining imaging and EPHIS together in the same experiments, potentially recording from cells at the site where you're imaging, things like that could be, very illuminating in this context.
Yeah, I agree 100%. I think you're right. I didn't mention that, but they do relate the fact that if you do single cell spike recordings, you do see a brief burst, let's say, of action potentials, and then you see the inhibition.
Yep.
And of course, the shunting inhibition, I think it's the most plausible explanation. I think it would be super interesting to record inhibitory neurons in V1 throughout this whole process. I'm definitely not an expert, but I know that the inhibitory network is fairly well described in the cortex by now. You have, different kinds of interneurons, PV, somatostatin, VIP. So I think it would be interesting to probe those cell types and see how they are behaving throughout this whole process. And the second thing that you mentioned, you're absolutely right. I don't think it's very often that people grab meaningful information from this neuropill signal. I think very often is kind of discarded as background noise or out of focus light that is maybe not reliable, or you cannot really know what it is. And I think it was a really good example that you can actually get meaningful data from this out of focus light, if you will, or this noise, that it's actually not noise, and it can show you, in this case, neurological adaptations to very specific stimuli. So I thought that was a good take-home message, that you don't necessarily need to stop at identifying your cells and defining your ROI and just take that. There's quite a bit of information coming from your entire field of view that you can source for meaningful insights.
Yeah, very much so. I agree. One question that came in, which I think you sort of hit on it, about the inhibitory neuron subtypes, but maybe there's a little bit more to say, was a question came in asking if the authors did any speculation about inhibitory mechanisms or cell types contributing to the observed differences between the EPHIS and the calcium imaging. And I know they do talk a bit in the discussion and elsewhere about some of these subtypes. But my recollection is it wasn't as simple as, the VEP is picking up, from, whatever excitatory neurons, and then the calcium signal is picking up more of this inhibition. I don't know. Do you have any comments there?
I think it's obviously not a very straightforward thing to disentangle because of first of all, because of the complexity of the interneural network in the cortex, in the location of synapses coming in. Again, I'm not an expert, but I know that this is fairly well described.
Yeah.
And then on top of that, you have, the inputs that are coming to these dendrites. So is the VEP picking up interneural activity, or is it picking up the activity coming from inputs driving the raw visual input, or is it picking up both of them? And at which point in time is the inhibition coming in? I think that's why, unless somebody records specifically from these classes of interneurons, it's very difficult to speculate too far on that side.
Yeah, no, it's true. Yeah, and I mean, who knows? The lab may already be, certainly in a mouse, studying these kind of phenomenon. It's very available to them to potentially start imaging specifically from these subtypes. So I would imagine that might be a path that they're taking. Yeah, and I mean, I think on that note, also just a comment that came in, that it would be interesting to look at other kind of aspects here, not just subtype, but they focus on layer 4 probably for quite a few reasons based on the history of the literature and this phenomenon. But it would be interesting to image in other layers outside of layer 4, understand kind of how the whole laminar circuit essentially is contributing to this, and because I guess with the PRISM probes, at least, like, they wanted to focus on layer 4, but one could potentially, even in the same animals, image from layer 3 and layer 5 and see what kind of differences one might see in supergranular and infragranular layers. Another comment, just that it might also be interesting to correlate at the single cell level with the somatic calcium activity that they looked at, sort of the degree to which cells, certainly there must be sort of variability in to what degree individual cells are reducing their response with the habituation. It might be interesting to try to learn again more about those cells, things like even just physiologically, right? Like what they looked at orientation tuning, but it might be interesting to look and see, is there something about the cells that show the strongest reduction in response? Are those the cells that are tuned to that orientation that's being presented? Or do cells with narrower versus broader orientation tuning show more or less of this kind of habituation of response, spatial frequency tuning, all the kind of typical physiological measures that people look at to try to understand, again, is this, while it's clear that there's a really specific orientation tuning aspect to this, it's still a little unclear, like, kind of what the subcomponents of the circuit are, and both physiologically and genetically that are sort of working together and contributing to this.
Yeah, definitely. And I think even to go beyond that, but kind of in a tangent direction, this is still a very, very specific, let's say it's a very controlled experimental condition, right? You have a very specific stimulus, and that's why you can potentially find the cells that are specifically responding to that. But of course, we know that in a freely moving situation, the particular orientation of the head or the position of the animal will change that visual field, right? It will change the orientation of the visual field. Yet, you still have the same phenomenon. You still have the VEPs happening. You still have the behavioral adaptation that comes with it. In that first paper that I cited in the introduction, you still have that behavioral responsive approach that then gets habituated. So it would be interesting to understand, let's say, to merge the two things, and understand mechanistically what's driving that plastic process, and especially how it handles a dynamic, let's say, closer to a real world scenario visual field, where you're not always having the same orientation or the same particular visual stimulus. You're changing your position, your internal state, if you will, right. And yet it is still happening, right? You're still very specifically picking up or particular stimulus that is familiar to you and you recognize it immediately. So it's quite interesting to try to get at these things, but of course you need quite sophisticated experiments to be able to assess these things as accurately as possible.
Right.
So it's up to the creativity of labs to start tackling these questions.
Yep. Yeah, and actually, I mean, it would, normally, especially in visual system experiments, animals are, restrained, and you want to keep everything kind of consistent in terms of their eye gaze orientation, things like that. But it would be interesting in this particular case. I would be curious to see using more of the free behaving paradigm where they've already shown the VEP phenomenon and that behavioral habituation, to actually watch that, in that dynamic situation, but using the calcium imaging in layer 4 as they did to look at that, I think would be a nice addition. Let's see. I think that might have been it. Yeah, I think we covered everything. So, with that, I guess if there's no more questions, then we'll wrap things up. So thanks to Giorgio again for walking us through this interesting publication. And thanks to everyone else for attending and sending in questions. We hope that you'll join us again next week when we'll be going over another recent circuit neuroscience publication. Until then, please stay safe and healthy. And hopefully we can all get back to our regular research activities soon. So thanks again, Giorgio. Appreciate it.
Thank you, Giorgio.
All
Goodbye.
Right. Yep. Take care, everyone.